Okay, we're back online. Let's get this thing back up. Here we go. So, first half was all about hormones. Second half is all about the nervous system. So the nervous system we're going to use to respond quickly. It has the ability to respond quite quickly. The response is short-lived, and we get on with things again. So that there needs to be a mechanism for turning off as well, clearly. The central nervous system, most people in the cycle know this better than I do, but the central nervous system is essentially our head here, our brain, with the central nervous cord, the spinal cord coming down here, our notochord, and that's where all the processing takes place. And this peripheral system is about the sensory nerves bring information in, and the motor nerves taking information out for in effect. You see afferent and efferent as alternative language for that, but it's the same thing. And there's also some interneurons that connect the afferent and efferent across the central nervous system, particularly through the spine. So we find these in higher order organisms. We saw in our planarian the other day a very simple version of this, but it was certainly developing. There was a network of nerves, there's a nerve collection, a neuron collection in its head, with lots of sensors for things like light and food, chemosynthetic sensation as well. So it needs to respond to those sort of things. Importantly, as I said here, is quick. We need a quick response. Just done that, so I'll jump that one. This is a diagram I used in my year 9, so I'm sure you've seen it. And it's misleading. John Wiley and Sons, hang our head and shame. No, so it's a wonderful diagram of this nerve cell for its purpose. It's pointing out how a nerve cell is different to other cells. But what it often doesn't do is allow students to realise that it is still a nerve cell. So sure, there's a nucleus there, but where are all the other organelles? Of course they're still there. That nucleus contains the DNA for this cell to produce proteins for its own maintenance of its metabolism, but also, of course, more importantly, DNA to make neurotransmitters to send messages along the neuron. So this should be ER and ribosomes and Golgi apparatus and lots and lots of mitochondria, because it's such an energy demanding process. So we do see the main points. So we've got the cell body here with its dendrites reaching out, and they could be reaching out to the back of the eye, they could be reaching out to another nerve. But there's our dendrites from our cell body. This axon runs down the length of the cell then to the axon endings, and these synapses. And this could be a gland or a muscle, or again another nerve at the other end. They're wrapped with myelin sheath, Schwann cells, which insulate and ensure that the message is quick and travels down this axon, not out into the interstitial fluid. We don't have message being lost at all. And they're the main differences we find in a nerve cell. The important part for us in many ways is this ending here, this synapse. So there's one axon coming in, the synapse is this gap between them. We've got neurotransmitters being released from a vesicle. That vesicle, of course, is broken up a Golgi apparatus. And the receptor sites, on the other side here, to collect our neurotransmitters and start the message down the next dendrite, down the next nerve cell. And you'll see here that once it's being used, that enzyme exists here to destroy this particular neurotransmitter. In some cases, the neurotransmitters are taken up again by the axon, so they can be reused. A lot of energy, a lot of chemical material has gone into making those neurotransmitters. So, you don't want them causing a repeated response, and you've got the shakes, but you also don't want to waste all that energy. So either they're destroyed or they're recycled, or destroyed and recycled, which is an important point. It's come up in the exam a couple of times in recent years, this idea of what happens when it's finished. What must there be in here to change things? So potentially an enzyme for destruction, or even a carrier protein to take it back to where it came from. I think I'll jump that for now. That's probably enough. This is the important part. This is the action potential. And I was talking the other day about the salty bananas. And you can see our potassium ions leaving the axon, so the axon's membrane. So our potassium leaves and sodium enters. And as the action potential, as the it's not really electricity, but as the electric charge runs down that membrane, you've got potassium going out, sodium coming in, and of course behind that, the sodium goes out again, the potassium comes back in. But you get a wave, this wave here, down the axon. So there's a threshold. You need minus 55 microvolts minimum for the stimulus to work. So if that doesn't happen here, this doesn't continue. So you can have failed initiations. If it's strong enough to go past the threshold, it generally needs to, well, not necessarily, but it'll jump quite high and then depolarize, come back below the resting stage and come back up to it. And that wave can be drawn up and down this axon as it passes through. So what's happening with that wave? Sodium channels open, more sodium channels open because it's gone past the threshold. Sodium channels close, potassium ions open, potassium ions close, and that runs along this axon. That's called the action potential. We'll watch Bozone's video because he does it really well. He talks about the salty bananas. So sodium chloride, your salt, potassium bananas. It's a nice little metaphor, but he does it, has a really good animation of how that works. So rather than stuff up trying to do the same thing, we'll watch his because it's very good. Well worth watching over and over again, actually. He does it really well. One thing he talks about in his is the idea of, if I just lightly touch the end of my finger, I feel that. And so that could be this little peak here. If I flick it hard, the peak won't be any higher, but the wave will be really tight. So back up. A really, really tight wave. Okay? Touch it lightly and the wave's slow. But he does that really well. So we'll look at him next class. So what other sorts of signaling molecules are there that work in our system? Well, there's these things called cytokines, and they're proteins, and they're part of our immune response. So they often stimulate the movement of cells towards sites of inflammation. So if you've got a cut or an infection, then lots of material needs to turn up and try and destroy any invaders. And we'll talk about that in the next section of the course. So we find them in a broad range of cells, and they're used particularly to attract other immune system cells to a site of infection to deal with the problem. So we've got cytokines. Pheromones, we touched on pheromones in the previous podcast when we were talking about ethylene. Ethylene is a classic that we know about working. We also know humans produce pheromones. All animals do. That's what animals use to mark territory, to use sexual attraction. Insects are great for, the ants live a food trail, and the ants run up and down that same trail. They're releasing pheromones to tell their partners where to come for the food source. All those sort of insects do the same sort of thing. So pheromones are a hormone, a protein, and they're used outside the body to, again, communicate with other members of the same species in some way. And the other part of this first part of the course is apoptosis, which is strange if you think, oh, it's just cell death, but it can be initiated in two ways. And of course, the initiation of it in each way is cell transduction. So it's worth talking about. So we can have what's called an intrinsic pathway. So the mitochondria within the cell sets off the process to destroy the cell, or we have an extrinsic pathway, which is the death receptor. And we saw that in action in that lovely Woodleigh.TV Apoptosis animation from Drew Barry, where we see this wonderful cascade. But again, that death receptor that is connects to the death ligand on the surface of the cell's telling the immune system it needs to die. Either it's aged and it's time to fall apart, or it's infected and it wants to be cured before the infection spreads. The death ligand sets off externally from outside, sets off the apoptosis pathway, but it ends up at the mitochondria, and the mitochondria finishes the process. So that mitochondrial pathway still gets involved. Or the cells decide its life is over, and intrinsically the mitochondria start the pathway and the cell falls apart. But it's all mediated by caspases, enzymes. We saw those caspases cleaving different proteins in that wonderful video, so worth another look. It is a really good little video. It's only a few minutes. It's free on Woodleigh.TV, so ad there for Woodleigh. Great work they do. However, there is a problem. Sometimes our apoptosis goes wrong. And my head's in the way there, isn't it, for that diagram? It gets me out of the way. Yeah, where can I hide my head? Over that corner? Yeah, that'll do. So in this case, here we're seeing Alzheimer's. So here's our healthy brain, and it's nice and pink and full of neurons and glial tissue. And here's our Alzheimer's patient who's obviously had Alzheimer's for quite a while, and there's gaps everywhere. So a lot of cells have been dying, and dying before they need to. So something has triggered their apoptosis pathway. So apoptosis in this case has gone wrong, and our brain is deteriorating. So it's a degenerative disease. This one here is cancer. And cancer is also when apoptosis goes wrong. But in this case, apoptosis gets turned off. So the cancerous cells no longer do apoptosis. And by no longer doing apoptosis, these growths become larger and larger. And of course, in this particular diagram, we've got a blood vessel around the lymph node there. And this little tumour is slowly growing. But these cells aren't doing what they should be doing. They should be part of the blood vessel. They're blocking the blood vessel here, blocking flow. Then certainly not vessel type tissue. And of course this part here is the important bit. As it metastasizes, it breaks up, it starts to spread this cancerous growth around. And these cancerous growths don't die. Because of course apoptosis doesn't work. And we'll talk a bit more about that in the next one as well. But that, I'm pretty sure, is it. It is.